Today has joined. Good evening, everybody. Mr. Lal Mohan Sangha is with us. Good evening, Mr. Lal Mohan Sangha.
Good evening, sir. Hello,
Thank
sir.
you. Yeah, thank you for accepting our invitation and being here. We will begin with the formal program. I now hand over the session to Maithili.
A warm welcome to all. I am Maithili on behalf of National Science Association, St. Joseph's University, welcoming you to the national webinar on Biodiversity Hotspots of India as a part of Call of the Wild 2.0, taking place over the course of 2 days with 4 wildlife biologists from 4 different regions with biodiversity hotspots. I would now like to welcome our speakers for today's webinar. Firstly, I welcome Mr. Lalmohan Sangha, who is a Senior Research Fellow under DRDO and will be speaking about the biodiversity of Indo-Myanmar hotspot. A hearty welcome to you, sir.
Okay. Can we start?
And
Oh,
our next speaker for the evening is Mr. Sachin Bhaskar, a research scholar at the Indian Institute of Science Education and Research in Thiruvananthapuram, who will be talking about the marvels of the Western Ghats. I would also like to welcome Dr. M. Jaishankar, Teacher Coordinator for NSA and HOD of Zoology Department SJU. I would like to invite Sir to share a few words with us. Sir, you are on mute.
I'm sorry. Am I audible now? Okay. Thank you. Thank you, Maithili. Uh, Welcome, Mr. Lal Mohan Sangha, the first speaker for this 2-day national webinar on biodiversity hotspots of India. This we are conducting from the Natural Science Association, Department of Zoology, School of Life Sciences, St. Joseph's University. Now that's the change in name from college to university. We had the herpetology webinar last year, and Lal Mohan Sangha was one of the presenter, and it was St. Joseph's College, and we are now stepping into the university mode. Uh, the Call of the Wild 2.0, uh, organized by the Natural Science Association, is keeping in mind to drive home the message on wildlife conservation. So that's the week-long celebrations and activities that are being planned by NSA. And this year's theme on the national webinar is about the biodiversity hotspots. We all know the 4 prominent biodiversity hotspots of India: the Himalayas, the Indo-Burma region, the Western Ghats, and the Sundaland. And interestingly, we have got speakers from each of these regions, representing each of this region. We have Dr. Meherin representing the Himalayas, and will speak about it. Uh, Mr. Lal Mohan Sangha is here, the Indo-Burma region, and Mr. Sachin Bhaskar will speak on the Western Ghats, and Dr. Sahu about the Sundaland. Uh, Dr. Meherin and Dr. Sahu will speak tomorrow. Today, in the first session, we have Mr. Lal Mohan Sangha and Mr. Sachin Bhaskar addressing this national webinar on biodiversity hotspots of India. Development, urbanization, pollution, disease seem to be major causes of concern to this planet's biodiversity. There are few recognized regions, 34 to 36 is what the number is put globally, are identified as hotspots, which have the two major criteria to categorize them as biodiversity hotspots is: one, their rich endemicity, and two is also they are threatened. India has 4 major that I mentioned, and these biodiversity hotspots almost globally take only 2.5% of the Earth's land surface, but account to almost 43% of the bird, mammal, reptile, and amphibian diversity. This is sufficient to tell why it is important to conserve these biodiversity hotspots. Let us know more about these biodiversity hotspots in India, the rich biodiversity that prevails in these four regions, the concern and the conservation initiatives that are associated with these regions. We are going to hear it from the speakers from the ground. The first session is here with Mr. Lal Mohan Sangha. Over to Abhishek to introduce the speaker to us, and we'll go ahead. Thank you.
Hello everyone, I am Abhishek Mishra, President Natural Science Association, and I heartily welcome you, Sir Lal Mohan Sangha, for national webinar on biodiversity hotspots of India. Sir Lal Mohan Sangha is currently working as SRF under DRDO project entitled Evolution of Snake Repellent against the Principal Venomous Snakes of Northeast India under laboratory conditions. He has a work experience as a field assistant under DBT sanction projects and had done work all over the Mizoram for the past 7 years. He has published more than 55 papers during the past 4 years and has also described 6 species from Mizoram during this time. He recently rediscovered the Doria's foam nest tree frog, Chirigellus dorige, which was last reported from India 109 years back. He is currently working at Munnal National Park, situated on the near border of India and Myanmar. He works under the supervision of Professor S.T. Lalrem Sangha, the most renowned herpetologist from the Northeast India. Sir, now I welcome you to the virtual stage.
Thank you. Thank you. So, can we start with the presentation?
Yes,
sir,
please
you can
go
start
ahead.
with the presentation.
So, firstly, I would like to thank Dr. Jayashankar for giving me this opportunity to present in this national webinar covering all the biodiversity hotspots of India, inside India. And today I will be talking about the Indo-Burma biodiversity hotspot. Since I am living in Mizoram, this is not a new thing for me. So, but mainly as a herpetologist, I will talk about more on the herpetofauna and other. I will also include some other wildlife aspects as well. So, as you know, my name is Lal Mohan Sangha and I'm the SRF in the DRDO project here in MZU, Mizoram University, and I am doing my PhD on the breeding biology of the Dutafinas chanda in Mule National Park. So I'm working very close to the Myanmar borders, just on the Champhai district on the east side of Mizoram to do my thesis. So let me start my presentation here. What is a biodiversity hotspot? A biodiversity hotspot is a biogeographic region that is both a significant reservoir of biodiversity and is threatened with destruction. The term biodiversity hotspot specifically refers to 35 biological rich area around the world which have lost at least 70% of their original habitat. But to identify a place as a biodiversity hotspot, according to the Conservation International, to qualify a hotspot, a region must have two strict criteria. It must contain at least 1,500 species of vascular plants as endemic. It has to have lost at least 70% of its original habitat. These are the 35 biodiversity hotspots all around the world, and in India we can see that Indo-Burma, the Sundarlands, which includes the Andaman and Nicobar Islands, the Western Ghats, and the Himalaya, are the 4 biodiversity hotspots of India. Himalaya includes the entire Indian Himalayan region, and that falling in Pakistan, Tibet, Nepal, Bhutan, China, and Myanmar. Indo-Burma includes entire Northeast India except Assam, and Andaman and Nicobar Islands, since the Andaman and Nicobar Islands fall in the Sundarlands. The Sundarlands includes Nicobar group of islands and Indonesia, Malaysia, Singapore, Brunei, and Philippines. Western Ghats and Sri Lanka includes entire Western Ghats and Sri Lanka. Introduction. Indo-Burma includes entire Northeast India except Assam. This is the same thing, so I won't repeat. And comprise the area of the Indo-Burman biodiversity hotspot is more than 2 million kilometers square. Six large mammal species have been discovered in the last 12 years. And recently, one new langur was also described here. So the large antler muntjac, the Annamite muntjac, the grey shank dog, the Annamite striped rabbit, the leaf deer, and the saola. These hotspots also host remarkable endemism in freshwater turtle species, most of which are threatened with extinction. You will see the pictures later here, which are present in my place, Mizoram, and other northeast region also, due to over harvesting and extensive habitat loss. Bird life in Indo-Burma is also incredibly diverse, holding almost 1,300 different bird species, including the threatened white-eared night heron, the gray-crowned crocuus, and the orange-necked partridge. So these are the mammals and the birds which are recently described from the Indo-Burma hotspot in the last 12 years. And this popalangur, if you can see in the bottom, it just looked like the the common langur, but this was discovered from examining the carcass and the poop of this popalangur from Myanmar. So these are the animals which are recently described from the Indo-Burma hotspot. This hotspot also hosts remarkable endemism in freshwater turtle species, most of which are threatened with extinction due to harvesting and extensive habitat loss. Bird life in Indo-Burma is incredibly diverse, holding almost 1,300 different bird species, including the threatened white-eared night heron, the gray-crowned crocuus, and the orange-necked partridge. So the hotspot original extent is more than 2 million kilometers square, and the hotspot vegetation remaining till date is 118,653. Endemic plant species 7,000 plus. Endemic threatened parts 18. Endemic threatened mammals 25. Endemic threatened amphibians 35. Extinct species 1. Human population density is 134 people per square kilometer. Area protected is 200, 235,758. Area protected in category 1 to 4 is 132,283. Next, plants. Knowledge of plant species within the Indo-Burma hotspot is uneven and hampered by socio-political divisions and taxonomic complications. A conservative estimate of total plant diversity in the hotspots reveal about 13,500 vascular plant species, of which about 7,000, the 52%, are endemic. Among the flora of the Indo-Burma hotspot are a wide array of orchid and ginger species. There are more than 1,000 orchid species in Thailand alone, and most tropical hardwood trees, including commercially valuable Dipterocarp species and Technona grandis. Now, we will look into the wildlife now. Before that, aquatic ecosystems are also under intense development pressure in many areas. Freshwater floodplain swamps and wetlands are destroyed by draining of wet rice cultivation, particularly in Thailand, Myanmar, and Vietnam. This is the species diversity and the endemism of different animals in the Indo-Burma hotspot. So the plant species is you can see the endemism. Plants 51.9% is endemic, mammals 16.9%, birds 5.1%, reptiles 39.1%, amphibians 53.8%, freshwater fishes 43.8%. Indo-Burma Biodiversity Hotspot is one of the most extraordinary hotspots holding decent amount of species described and yet to be described. For example alone, in the field of herpetology, several species of reptiles and amphibians were described recently from northeast India in the name of a single laboratory, that is the RLab, which is the Developmental Biology and Herpetology Laboratory. So these species are such as Cyrtodactylus benkoyai, Cyrtodactylus aaronbaueri, Cyrtodactylus carsticola, Cyrtodactylus lungleiensis, Cyrtodactylus siahaensis, Cyrtodactylus agarwali, Cyrtodactylus virectensis will come out, Trimeresurus mayai, Herpetorius moorland, Ichthyophis benji, and so on. So I will show you the pictures of these Cyrtodactylus here. These bentod geckos, which are three bentod geckos, are pasted here in this slide. The first one is Cyrtodactylus aaronbaueri, the second one is Cyrtodactylus lungleiensis, and the third one being the last one at the bottom being the Cyrtodactylus carsticola. Here is the Trimeresurus mayai, which is recently described from Champhai district in the eastern side on the Manipur border of Mizoram and Meghalaya in the Umroi military station, which is located in just on the outskirts of Shillong, which is the city of Meghalaya. Here and below is the Ichthyophis benji, which I recently mentioned, which is recently described from Mizoram. Sirig's dory was again rediscovered from this hotspot after it was lost from Indian science since 1912. Anandel 1912 reported this from Arunachal Pradesh, which was the first report of this Sirig's dory. But until 2020, this species was not found again in India, was not described. The natural history, the behavior, the breeding biology was not again described. But luckily at Puareng Sanctuary, which is located inside Mizoram, which is inside the Indo-Burma biodiversity hotspot, we rediscovered this species again. So other rare species from the Indo-Burma. The first one is the Blithia muifang. Blithia muifang is uh, described from Muifang Community Reserve Forest, which is only around 50 kilometers away from Aizawl city, the main city. The second one in the middle is the Theldharma baibungens. This is also a rare frog, which is prominent in the Thailand region and Vietnam region. The third one, this soft-shelled turtle, is the Amidia ornata jongli, which is recently 2022 was described in Bangladesh. So one story about this is that we were writing a paper on this Amida ornata jongli. We were describing this paper, combining our manuscripts and restructuring our manuscripts, and during that period, this species was described from Bangladesh. So we were very unlucky in describing this species. And the frog at the bottom is Theldharma nagalandensis. It was described by Orlov et al. From Nagaland. But since then, it was not even seen in different parts, in Nagaland also, in the main paratype, and holotype locality also. But after this, description, we are the first one to be able to capture this image and even capture the specimen for DNA sequencing. It was captured from a community reserve forest. And the amazing thing about this is that the call is like a chirping sound of a bird. And it is only found in Mizoram also. Since we have explored most of the different parts of Mizoram, this particular frog is found in a particular tree, which is a cinnamon tree. The other species is the Trimeresurus popiorum. This also is found in the northeast abundantly, like in Meghalaya, like in Mizoram, Manipur, etc. They are found in only high elevational places. This is the first record of the Dutafinas chandai from Manipur state, northern India, with update information on its distribution and natural history. So this also, Dr. Finas Chandai also, it was described from Nagaland, but was later reported by Lerem Sanga et al. In 2019 from Mizoram, and was later again reported by the Simpson et al. From Manipur. So this species is also only found in this Indo-Burma hotspot, and this species is my thesis, so I will elaborate more on this. This species is the only toad which is found to uh, come out to breed only for 3 nights per year in the streams of Moorland National Park and Muifang Community Reserve Forest. They are distributed only in high elevations only. This is Haplobatrachus littoralis. Before we think that this species is the common tiger frog, which is the tiger bullfrog, which is the Haplobatrachus tigerinus. But later on, from DNA barcoding, we found out that this species was a species which is quite endemic to the northeastern India, the Indo-Burma hotspot, and we published this in the mitochondrial DNA part B also. uh, we describe the we also in our study we used to describe many frogs. We used to describe their natural history and many biology, and here is a paper where we describe the oral morphology of the Fajarvaria multistriata. This was done in Nehu, the safe. uh, so we published this in Sauria, a German journal. Uh, Here in Mizoram, in the Dampa region also, the Bangladesh side, but which is included in the Indo-Burma spot, we did an annotated checklist of amphibians in and around Dampa Tiger Reserve also. So the results were a total of around 28 species. Chamdil National Wetland is also located on the east side of Mizoram. So there are many things which are not discovered and which are still yet to be discovered. But what I can say is that uh, particularly Mizoram has a very good team of herpetologists, not only our team, other teams also. It is the most explored place for of herpetologists, because the native people here like us, who are staying here, studying here, are doing uh, the main people here like us, the PhD scholars and the research scholars, the MPhil scholars and other students are doing their works in Chamdil National Park throughout Mizoram. So here in 2010, we described a new species of Leptobrachella. Before it was named as Leptobrachella. What we have to know about amphibians is that their names always change day by day depending on the authors. Okay, they always change the name of the genus, but the specific name never changed. This is the Leptobrachella tramdil, which is described from Mizoram in the year 2010. They are found mostly in higher elevations, but in moderate elevations also we found this specimen, but mostly are found in high elevation places, localities. Here is the new species of Ichthyophis benji, which is described from Mizoram. This was in 2021. We described this species by counting the annuli and doing a morphology, molecular work on this. So this is the the fourth species from the Northeast India. So Kamei et al. When working with Biju, when they are working in the Delhi University, they are from the environmental science department. They came to the northeast into the Indo-Burma biodiversity hotspot, and they found that they discovered or they described 3 new species of Ichthyophis also, which are named Ichthyophis kumzai, Ichthyophis mustachius, Ichthyophis sandiniu. This is Ichthyophis benji, and these are the author lists. Chikila, which is an anuran, was also found in Mizoram in the Kolasip district. Kolasip district is close to Assam, but this is still in the Indo-Burma hotspot. So this was done by Kamei and Biju, Kamei et al. So in Mizoram we found Chikila fulleri. So these are some species which are described and which are rare for the from the mainland Indians also. So the first one, the snake, is the Mizo rain snake, Smithopis atemporalis. And the second figure, this soft shell turtle, is the Nilsonia nigricans. The most interesting fact about Nilsonia nigricans is that it was listed as endangered from before, but listed as extinct in the wild from way long way back, from way back. But recently it was again found in the Brahmaputra and some temples in Assam. But now here in Mizoram we have an abundant distribution of these species. See, they are endangered till now, critically endangered species, Nilsonia nigricans. So under this, we have an orchid. The orchid is Papilionati vandaram, which is found in high elevation places like Shillong and Mizoram, Manipur, the high elevation places. These species of plants, orchids, are also listed as endangered till date. So here on the left side of the orchid we have a frog. These frogs seems to be different in morphology, but if we study the molecular data, we published this last year 2021. This is the Nasutixilus jardini, the tree-hole frog, which is found in the Indo-Burma Biodiversity Hotspot only. Shillong, Mizoram, Burma. So the first one is from Mizoram, the second one is from Shillong, Meghalaya, and the third one is from Myanmar. So this species is having different colors and different morphometric form, but after studying the molecular works, we can conclude that this is all the same species. So these are the other rare species. The Dutaphrinas chandai, as we already heard, and the Odorana moflangensis, the one with the dorsally green coloration, which is found abundantly in Mizoram, Manipur, and Meghalaya. The third one is the Leptobrachila tramdil. The fourth one is the Lepeltis venatus, which is found in the Vietnam, Thailand region, and even in Mizoram. But we haven't heard any reports of this from other districts of Mizoram. Districts other than Mizoram. The fifth picture of this frog on a white background is the Xenophris cerchipai, which was described in 2008 by Matthew and Sen. So this also was found in Mizoram in Serchip district, which is Serchip district is located on the southeastern part of Mizoram. And the last picture of this gecko, Tychozun, is to be described yet. This will be named as gecko Mizoramensis. So it is described from the farther south side of Mizoram, Longkai district, and it has a very peculiar characteristics. They are called gliding geckos. It was placed under the genus Tychozun before, but currently it will be placed in gecko species. So they have flaps on the legs, on the hind limbs, and even on the forelimbs, and a patagia on the side of the body also. Excessive skin formation, which is used for gliding. So the first picture on this slide is the Megrophis awu, which is recently described in the year 2018 by Stephen Mahoney et al. Along with uh, his wife Swam Relu Kamei, which I recently mentioned, describing the ichthyophis and all. The second one is the Polypetades mutas. This also is only found in Vietnam before. No Indian records. As soon as a new paper will come out, a new country report paper will come out on this on the International Journal of Herpetology. And the third one, I mentioned the third one, though it is abundant in also in the Western Ghats, because this third frog, the greenish frog, brownish and greenish frog, was named after the Indo-Burma Biodiversity Hotspot. It is known as Amolops indo-burmanensis, which belongs to the family Ranidae. And the first picture on the bottom is the Herpetorius moorland. This Herpetorius moorland, it was described in the year 2022 by Amit Kumar Bal et al. This is described from Moorland National Park, the place where I did my thesis, and it's very recent to science. This is very new to science, Herpetorius moorland. So The next species is Calotis gisleri, which is described from the Myanmar, but we have also reported it from Mizoram and Manipur, both of them. Before it was mistakenly identified here in our laboratory, but after the molecular data has come out from Myanmar, we can confirm that this is the Calotis gisleri. And the last picture of this slide is the Kalotis Zhou Lai King. Zhou means Mizoram, Lai King means lizard. This is described from Mizoram by Larongnunga et al., who works extensively on herpetology also. And this is also found only in one place in Mizoram, which is Muifang Community Reserve Forest, and nobody has reported this outside of Mizoram. We'll move on to the next slide. So we compiled the critically endangered species of turtles and tortoise of Mizoram here of the Indo-Burma Biodiversity Hotspot. Since we are located in the hotspot, and since I am studying inside this hotspot, I am concentrating more about my place and these endangered species as well. As I mentioned, Nilssonia nigricans was described to be extinct in the wild, but here in Mizoram we found it, and we already published papers on the reports of this in the wild. In Doptesuda elongata, this is the uh, this tortoise is also placed in the critically endangered under uh, IUCN. So this tortoise also we have abundantly in the campus also in here in the campus also, and we have a turtle terrarium in our lab since we are working on a project for the DBT. So we have more than 30 of these endangered Indo testudo elongata, and we breed them successfully. Last week only we have 5 hatchlings which are very in good conditions. They feed well, and we plan to release them in the wild as soon as possible. So the Lysimis punctata is also the soft-shell turtle, which is found on the northern side of Mizoram. Amida, Ornata, Zhongli, I have already talked about this. Kaura Mahoti, also endangered. These are also found in the Pualing Wildlife Sanctuary, many other sanctuaries also in Mizoram, but are not yet reported from other places of the state. Chrysodorsa otai. I placed this Chrysodorsa otai because it was again described from Mizoram at Sairep, yeah, in Lunglei district in the southern side of Mizoram. So Rohanic Zilla senapatiensis on the bottom. This frog is very unique, and the chirping sound also it quite resembling the sound of a dropping of water. So this is found high up in the trees above ponds in Mizoram. Throughout Mizoram, to be they are found to be present in almost in every part of Mizoram. And another species of Rohanic Zillas from Mizoram will also come out as a new species by maybe by next year.
Mr. Lal Mohan Sangha,
Yes.
sorry to interrupt. There's that window showing meeting now. Can you minimize that? It's covering the images, right?
Okay,
Yeah,
sir.
thank you. Thank
Thank
you.
you, sir. Sir, you can hear me properly, right?
Yes, it's audible. It's
Okay.
clear and the flow is good.
Okay, thank you, sir.
Thank you. Carry on.
So I also put some uncommon species which are not found in the mainlands here. In the mostly they are not found in the mainlands. Example, the siro. The siro is the state animal of Mizoram, and number two is the binturong. The binturong is a bear-like from the bear family, which is omnivorous. It can eat both plants and meat also. They are omnivorous, so they are found high in the trees also. And this third picture is the clouded leopards. And according to scientific studies, Mizoram has the most population of clouded leopards in the whole Southeast Asian. Among Southeast Asian population, Mizoram has the most number of clouded leopards inside us. So this photo was captured from Murle National Park. There are lots of photos from our trap cameras. So uh, maybe I can show them later. And the picture on the bottom left is a bear. This bear is a sun bear. It is found in this Mizoram and the Myanmar side of Myanmar side, but mostly in India, the species which are found are the Himalayan black bears, but not these sun bears. The Hulok gibbons, the one which is brownish color is the female, and the blackish color is male. So the first picture here is the this is also very common in the Indo-Burman biodiversity hotspot. The clouded leopard is very common here. Is very common here, and the jungle cat also is quite common here. We have a fishing cat also. We know that many population and projects are going on fishing cats of Odisha, but Mizoram have plenty of populations. We don't have to preserve them till date. Their population is still high in this area. And the third picture is the slow loris. So this is the only venomous mammal. So I will tell one story on this. At Moorland National Park, when they are doing Zoom cultivation, we used to follow them for some herpetological works like herpetological surveys and all. So there is an incident where we encountered that when felling of trees happens during Zoom cultivation, they used to do slash and burn. During that slash and burn, there is a big tree with a hole on the top. So when my friend's grandfather fell that tree, this slow loris came out and beat him. And in 30 minutes, he has stomach pain, uh, loss of consciousness. He's having nausea. He's vomiting. And after we gave him paracetamol and some Avil injection, antihistamines injections and all, he recovered after only 1 day of staying there in a hut in Murulan National Park. This is happening in the buffer zone of Murulan National Park, the fringe area. And the fourth image on this is the Asiatic golden cat. We have a number of, we have the, what will I say? I can say that we have the most number of Asiatic golden cats here in Mizoram. And this paper on the small carnivorous mammals of Mizoram will come out soon from the Mulan Sanctuary and the Dampa Tiger Reserve, which is the biggest tiger reserve in biological wildlife reserve in Mizoram. So this last picture is the picture of the Chinese pangolin. Lots of illegal pet trade is happening here in Mizoram, and these scales, the scales of this pangolin are usually sold on the Burma into Burma. There is more than 1 kilogram of scales in per individual of this species, and they used to sell this to the Myanmar people at a rate of around 25,000 per kilograms. So uh, in one animal also, if they found more than 1 kilogram, so they can find it. They can find it. The local peoples here knows how to find these animals very easily by digging their holes and all. And since it's available for easy cash, and since they can sell it easily, these poaching of these scales is happening here in Mizoram. But since these people are living in remote areas and very under very dense forest, they are very hard to catch also. But I have found some incidents of selling of these pangolin scales from Mizoram. So the price is around 25,000 to 30,000 per kilograms. So we have Indo-Burma Biodiversity Hotspot is very rich in the bird fauna as well. So these are some pictures of ours of our lab, which I mentioned here, which I put here. So the endemicity of these birds are also high, and lots of birding is done here in Mizoram. Lots of researchers came out from the US, from the UK, and we have such a good diversity of birds. Since I'm not not very good in birds, I will not talk about this much. So I want to show you a video where you can see the scenario, the landscape, and how our terrain is in Mizoram, and some animal footage also here. So, Indo-Burma biodiversity hotspots in India, especially Mizoram, Nagaland, Manipur, Meghalaya, Arunachal Pradesh, These most of our places are hilly areas. So from this video, you can see what our conditions look like. And this is the video from Mulan National Park. So our terrain is what I want to elaborate on this video is that since our terrain we have a very mountainous terrain in our places. It's very hard for researchers also to do our research here. We have lots of challenges, lots of steep falls, steep waterfalls, and different other challenges also here. So it's quite difficult for researchers to work here also. You have to be very uh, passionate and loving for wild animals, loving for your land, and loving for everything in the world. So it's very hard to work in this kind of terrains more than the plains. So I will show you one picture of this small video. I will show one small video of this. Oh, this was compiled by our laboratory. So the challenges and the threats of this Indo-Burma biodiversity hotspot. The first one is deforestation. Deforestation occurs because of many reasons like urbanization, like building of uh, construction sites in the forest, building of resorts, and many other things. And the main problem is due to shifting cultivation in Mizoram. In Mizoram, shifting cultivation is the uh, major problem because it is the main source of income for the villagers, and there is no specific ways in which shifting cultivation can be prevented. So the government needs to uh, come up with a new idea to what will I say? Yeah, a new idea to change and to uh, this this type of income. So the villagers needs a much more appropriate income other than shifting cultivation to quit this shifting cultivation. So forest fires often happens due to the slash and burn process, which is done for shifting cultivation. Once a land is used for cultivation, then it is not available for cultivation for the next year. So every year in Mizoram, in Manipur, in Nagaland, shifting cultivation is still uh, practiced, and it's still one major cause of deforestation of this area, of this hotspot. Even in Myanmar, the Chin Hills, the Tahan terrain, lots of shifting cultivation is going on. So this is the major problem for the loss of biodiversity in this hotspot. The second one is hunting and poaching. Hunting for trophies, poaching, and hunting for consumption of wild animals for the villagers is still very common here in this area, in this hotspot. Most of the people here living here are tribal people, and development is does not occurs in a fast pace. So hunting and poaching is still very common in this area. Mainly in Mizoram, they don't sell these trophies, but mainly they consume these wild animals for their main source of protein. And this is the scales of the pangolin which I mentioned, which is sold to the Myanmar area. So poaching is very common from the Indo-Burma biodiversity hotspots. It came through Mizoram, entered through Silchar in Assam, and is widely distributed into the mainlands. So these are the scenarios which is happening in Mizoram. The Champhai district, the Zaukothar site is the border of Myanmar, is the main road which links Myanmar to India. So at that place, the customs office and the forest departments capture lots of these poaching materials, like the even iguanas, pythons, ball pythons, even kangaroos is brought from Myanmar to be transported to different parts of the mainland Indias. So lots of exotic birds are captured here, and the main reason because of this is again because of easy money. Let me say easy money. So these exotic animals, which are exotic animals, this is actually native for the Indo-Burma biodiversity hotspot. But since it will come out of the hotspot and go to different parts of the country, even maybe outside to foreign countries, so this will become exotic to that place also, and it's very harmful. We all know the what harmful effects these exotic animals have on the native animals. Soil erosion. Soil erosion is also one of the most tragic challenges which we face in Mizoram, in Nagaland, in this Indian region of Indo-Burma biodiversity hotspots. Since erosion happens in mountainy areas, since all our terrain is mountainous, we have a good problem of a very huge amount problem of soil erosion here, and which leads to deforestation and siltation of the rivers, which affect again the aquatic habitats of this area. These aquatic habitats, we have lots of fishes, native fishes, and even let me say the introduction of exotic fishes like these fishes for consumption, such as the common carp, the rohu, grass carp, this kind of carps. This also is happening inside Mizoram, inside Nagaland, and even in Manipur for conservation. They say it in the name of conservation, but what they don't know is that this release of exotic animals to our land is very harmful. The competition between exotic animals and native animals is so high, and the survival the survival
One minute.
survival of
Sorry
these.
to
Yeah.
interrupt. Participants, please turn off your camera. Please ensure your camera is turned off.
Yeah.
So
Okay, let me go.
yeah, thank you. Please
Okay,
proceed.
let me continue. Lots of exotic fishes like the carps and these grass carp, rohu, raw mirga. Lots of them are released in our rivers. So the competition between the exotic and the native fishes also it's happening, and we have a declining population of exotic fishes, of native fishes in Mizoram as well as Manipur, Nagaland, and even Arunachal Pradesh also. This also even happens in the Myanmar regions of the hotspot, and these have to be fought. It is because of the this happens because of the what so-called conservation, but this have to be stopped immediately. Urbanization is also one of the threats and one of the most important cause of deforestation in these hotspots. So the first picture you can see, this is Aizawl area. The second is Imphal. The third one is Dimapur, and the fourth one is Shillong. So you can see the amount of land it took from being a pristine forest. So every people knows that urbanization is the main cause of deforestation, which again leads to the threats of the wildlife biodiversity, and again leads to the threats in biodiversity hotspots in India. So aquatic fauna poisoning is an unnecessary means of fishing. This is also practiced in all parts of the states. In Mizoram also, we have more than 10 rivers, 10 major rivers. It is also practiced in every river. No fishes are there to be caught like pristinely, like in a correct method, like what will I say, with nets, with throwing nets. Nobody practiced that. So what they did is they just poisoned the rivers, the streams with chemicals which are poisonous to fishes. Not only fishes, all the aquatic habitats such as crabs, such as snails, uh, eels, etc. So we have a very prominent decline in our aquatic habitats. Even in Manipur and Nagaland, the use of this native plant roots, also they used to crush the roots and squeeze out the juices from the roots and put in, they used to use it for catching fishes, but that doesn't kill only the fishes. It affects our tortoises, our turtles, our crabs, our shells. Lots of these unnecessary means of fishing is happening in Mizoram. And here also, lastly, I will point out that this kind of uh, dynamo fishing is also practiced in Mizoram. They used to electrocute the fishes and other aquatic animals in the small streams, in the main rivers. And this first picture also, they are caught in the Tuirini River. So the forest department will implement the WPA Wildlife Protection Act 1972, and they took action against this, and they were all sent to jail and with a fine of more than 25,000 rupees. So lastly, for my presentation today, I want to show you a video on how these people even in these states, even in Myanmar, even in Vietnam also it's happening. All the states of the India, all the states in India which is included in the biodiversity hotspot, Indo-Burma biodiversity hots, use this kind of fishing techniques, which eliminates lots of many numbers of fishes. And this is the place. The second picture is the place where we found the Nilsona nigricans, the critically endangered turtle from India, which is very important because since they are listed as extinct in the wild and later was recovered from Brahmaputra and Mizoram only. So this happens in Mizoram. If this happens in Mizoram and other parts of the world, what will happen to our wildlife? You have to think about that. So here I will play this video and you can imagine what the effect will be. It's a very short video. So let me play this video. And this is happening just in front of our eyes. And the one who throw this bomb, this is called a gelatin bomb, which is used for mining of limestones and this construction boulders and etc. So they throw it in the river. They join with more than one stick of this bomb, of the powder, gunpowder of the powder, and they throw it in the rivers, the hydel projects. So our declining fishes scenario can be understood from these kind of videos. So what I want to say is that the person who is throwing this bomb is also a forest guard from this area. This is happening in the Pualreng Wildlife Sanctuary, but since we don't know him, we told him to stop. And yeah, he stopped. This is taken with a 600 mm lens. Okay, so we are not near him to tell him directly that this is not good, this is not good for our wildlife. And since you are a forest guard, you have to guard this forest. This is taken with a 600 mm camera, so it's more than, I think it's more than 1 kilometer. So I already cropped the video, that's why it's quite unclear. So this is the scenario happening in Mizoram. So thank you.
Thank you so much, sir, for sharing such a wonderful presentation with us. Everyone present here appreciate your presentation and is enlightened with your talk, sir. Thank you so much, sir, again.
Thank
Now
you.
the Yes, sir. Now the talk is open for the question. Any question, my dear friends, can ask to sir.
Mr. Lalmohan Sangha, this is the issue with most of the biodiversity hotspots or protected
Yes.
areas. There is
Yes.
always a indigenous connect Okay, there's a
Yes.
link to culture in terms of food or celebration that you make use of the resources, may it be wildlife or plant and other thing.
Yes.
How to make indigenous communities refrain from, like you said, to avoid hunting, poaching, or using them part of their cultural thing, and give them an alternative, like we saw with the hornbills beaks being converted into wood and plastic in Arunachal. Any
Yes.
such awareness initiatives to reach out to the communities to create
Yes.
awareness?
We conducted many awareness programs. I have some video also. If I can share, I will share it again. I think I still have time. And what I blame is that the government here in the northeast is not as functional. It's not. Let me just say, keep it like this. It's not as functional and strong as the governments in the mainlands. So the government needs to propose new plans, new schemes for these villagers and these indigenous people, so that they can abandon these uh, practices like fishing, which they rely on income for, as and especially the shifting cultivation. See, we don't have any other alternatives. So this is the main problem which is still happening in the hotspot. So the government has to implement new plans and new policies for these indigenous peoples and these villagers. So I will show you a small video. Let me share my screen again, sir, please, if you allow.
Yes, please go ahead.
Okay, I will share. You can see my screens, right?
Yes, sir.
So this is our documentation on the Tampa Tiger Reserve. So what is done is that this shifting cultivation is going on. The number of shifting cultivation is going on in these areas. The slash and burn. They slash, they slash off the forest and they burn it for cultivation. The burning of these ashes are used as fertilizers for the crops to grow. Okay, hence are we the trap animals and trapping of animals is still very common. Hunting animals is very common. But what we did is that, see, so we used to under the sanction of the Wildlife Trust of India, Noida, that they gave us a sanction. This sanction was mainly to rescue snakes and to work with the snakes, mostly on snakes. But from that money, from the remaining money, we used to organize some awareness program on biodiversity conservation and livelihood program. So we used to do this to many villages. Right now we have went to to all of the protected areas in the almost all of the wildlife sanctuaries here in Mizoram, uh, which are all in the Indo-Burma biodiversity hotspots, and we used to explain to them, uh, community by community, we have to gather them in hosts. But the one big problem with financing again. If you want to call people from the community, they all have works in their farmlands, in their cultivation lands, in their lots of daily work is going on. So if you want to gather the lots of crowds, the villagers, you have to give them something, just like t-shirts, like fruity, something to drink, something to eat. So we have spent more than 5 lakhs this year only also for awareness campaign for conservation of wild animals and even wild plants also. So we have spent more than 5 lakhs only this year also, sir. So what we also did, sir, I can stop my sharing of the screen. What we did is also with the help of ASEP, we used to make an awareness video. I can share the video later. Maybe I can share it personally to you also. How bad this can affect our environment. Even our seasons are changing. The rainy seasons have shifted lately, which affects all the wildlife, the anurans, the reptiles, and the declining population happens because of this also. So we are still trying our best to stop this kind of activities in our state, Mizoram, specifically, sir.
Thank you. Thank you for that explanation. Yeah. Then going harsh on the government, I believe it's a tight walk for them too,
Yes.
to balance the aspirations of the people, at the same time look into initiatives on conserving the wildlife. I believe that's also a conundrum everywhere. Thank you for this presentation. Thank you for accepting our invitation to represent and give
Okay.
us
Thank you so much,
details
sir.
on the Indo-Myanmar biodiversity hotspot. I believe there is a question in the chat box. Will Abhishek moderate? Or to Abhishek?
Yes, sir. Sir, there is a question with a photo. If you
Okay.
can see the chat box, sir, there's a question that, sir, what is the name of this animal and what does it feeds on?
Wait, okay. I am not able to open my chat box at the moment. Can you just wait? Okay. Show chat box. I don't. Okay, I'm connected again. Sorry, I'm not able to see the chat box. So can you please find me? Uh, can you please direct me to the
Yeah,
chat
yeah.
box?
I believe they have answered it. So the answer is done of the pangolin and
Okay. Okay.
predominantly eating on.
Sir, I have a question, sir.
Okay.
So, like in our college also, and here in the meeting, there are many new like beginners for herping and birding, sir. So, if
Okay.
there is any new herper, a beginner trying to
Yes.
identify any species, so what
Yes.
the step he should follow to identify any species of any reptile or amphibian, sir?
Oh, these days, there are many works on the molecular data as well as morphological data also, the meristic data. So mainly what you have to do first, the first step is to have a permission from the state environment and forest department. The second step is to do an opportunistic survey. Opportunistic survey will bring you snakes which you collected, and after that you can do the morphometry works, whether it's snake, it is anuran, or whether it is what will I say, skink, lizard, or turtles. What you have to do is you have to work on the morphology. So firstly, after working the morphology, you cannot conclude that and identify the species, because India alone, even these hotspots, they are very rich in the, what will I say, the species diversity. So there are many cryptic species which are not identifiable just by looking at the morphology, which needs molecular work to be done. So there are lots of complex species, a group of cryptic species, which is called as complex species. Okay. So what you have to do is that you can find lots of data, old data, and which are studied data, which are available in researchgate.net, etc., and other sources. So by comparing them, and with the help of other some renowned herpetologists also, like Vishal Santra, S.D. Biju for frogs, he's called the frogman of India, and what you can also do like Whittaker, Giri. There are lots of good herpetologists here, and mostly the easiest way to do is by molecular identification. So in molecular identification, after you generated the phylogenetic tree, it's very easily identified. From that, before it was studied from the morphological level to the molecular level. So this work is going in a flow, in the forward direction. But now after we have molecular study, you can go backwards now. So the space for the study is more or less. So did you get my point?
Yes, sir. Thank you so much,
Yeah,
sir.
it is very easy from that now. Okay.
I believe just to add on to that, Lal Mohan Sangha, this is a forum that we have launched, the Josephite Forum for Herpers, where we
Yes.
go weekend field visits, looking
Yes.
out. There is also one for birding, one for herping. So they
Yes.
wanted to know as beginners when they get into field, set their foot out,
Oh, oh,
what is
oh,
that
okay.
they look for in nature.
Okay, okay, okay.
Everything is new to them. Yeah, that way.
Okay, okay. I will answer. I will answer to that. I'm very happy to get that question. So, when you get in the field, there are snakes which are nocturnal and there are snakes which are diurnal. There are frogs which are mostly all frogs are nocturnal. So the best time to go out is at night. So after this dawn till around 2 a.m. you can get number of snakes and frogs. And there the place where you have to look for is you can look for under cover, under shaded area, and even swamp areas, the place where there are a little bit of water and frogs. You can easily find frogs in stream, which will lead you to snakes, because snakes mainly eat frogs and other mammals. So the frog eating snakes, you can easily find it there. And but you have to be very careful, you have to be very cautious about your environment. See, one funny story is that when I was doing my survey in the Tokalo Wildlife Sanctuary, which is located in the southernmost portion of the Mizoram, since Mizoram is enclosed with Myanmar from the south and the east side and south side, we have Myanmar border. So at the extreme end of the Myanmar border, I did my harping. Suddenly, I was looking at the streams, try to collect as many as frogs as possible, since we do a DBT project as a field assistant. When I work there, when I put up my head and look forward, there was a Himalayan black bear in front of me. But since we were around tree, uh, we stood still there, retract slowly. Then that's the funny thing which happens now, the closest encounter to a Himalayan black bear. But you have to be very cautious about your environment as well also. You have to put your torch light on every time. When your torch light shines, where your torch light shines, you have to put your eyes there, and you have to be very what you use is mainly visual encounter system and acoustic encounter system for these surveys. Visually you can see, and through your ears you can hear. That is called the acoustic encounter system. So mainly those two are to be keep in mind for this herpetofaunal work which you are doing there.
And sir,
And I
timing,
thank
sir.
Sir J for I know that he's the person who is initiating all of this. So I thank him a lot for these small resources. And later on also, we both Sarjay and me, will organize a program in which we will tell students how to write and how to look for papers, how to write a scientific paper and how to publish it. So we will make some task on that. We will take some task on that. So uh, that will be in the future. So it will depend on what Sir Jay's thinks. Okay.
Yeah, yeah, thank you. You already laid foundation for our next webinar. You have been very helpful in that regard. Thank you for accepting our invitation for this webinar, and also readily willing to guide our students on writing. Scientific communication is must. Collaboration is the order of the day. And you got to that old adage of whether you publish or perish. You got to document things. Therefore, thank you for being there to help us. We'll soon reach out to you, work out on the modalities on the scientific writing component webinar. Thank you, Lal Monsanga. Over
Thank
to
you.
Abhishek.
Thank you so much, sir, for the presentation and the question and answer sessioning, sir. Now I welcome Selva Priya to introduce our second speaker.
A very good evening to everyone present here. I am happy to introduce the second speaker for today's national webinar on biodiversity hotspots of India, Mr. Sachin Bhaskar. Mr. Sachin is a research scholar at the Indian Institute of Science Education and Research, Thiruvananthapuram, under the supervision of Professor Hema Somanathan. For his PhD, he studies the plant pollinator interactions in the Agastya Malai Bioreserve. He also works on floral traits and understands how they act as signals in indicating reward status in flowers. Welcome, sir. It's a pleasure having you with us.
Thank you. Good evening to everybody. I'm very glad to have been able to join you to give this plant-centric talk of mine in the Western Ghats of India. I'll share my screen. The moderator can just help me know if everything is fine. Is it fine?
Yes, sir. We can see your sharing.
Okay. All right, then I think I'll start with my presentation without wasting any time. And thank you very much for that introduction. And what you did miss out is I was also a BS MS from St. Joseph's College, and I just passed out in 2020. So I'm also a Josephite. Yeah, okay. So today's talk is going to be about the Western Ghats. And since I'm a botanist and since I'm a plant ecologist, I'll be focusing a lot on plants, and I'll be telling you the various aspects of plant research and all the allied subjects with respect to the Western Ghats. So this is exactly what my talk is going to include. I'm going to keep it very short, and yet I'm going to make it I've tried my level best to pack it up with a lot of information, which I'm sure you guys would not have studied because these are works from my own lab here in the institute. So that is one thing. And these are the five major aspects that I will be looking at today. And these are the things, these are the lines that I'll be running with. I just have two things in my mind for the presentation. One is to make you feel awe about the Western Ghats and its biodiversity. And the second thing is I want you to uh, start loving plants equally. These are the two major things that I'm running with for today's presentation, and I hope I'll succeed in that. So, the first thing is we all know about the Western Ghats. These are parallel, long chain mountains that are running parallel to the western coast of the Indian Peninsula. But what we actually may not know is how they actually were formed. Now, we all know that there was a huge continental mass called as Pangaea, and it was somewhere in the 200 million years ago that this Pangaea broke down into what is called as the Gondwana lower land and the Laurentian upper lands. And then so in this, if you were to follow my cursor, you will know that the South America, South Africa, India, Seychelles, and Madagascar, all of them were components of Gondwana land in itself. And then later down the line, we find that from African plate, the ISM complex, which is the India, Seychelles, and Madagascar complex, breaks apart from the African plate. And this happened somewhere 130 million years ago. And this was believed to be the birth of the Western Ghats because the chipping of this ISM complex has formed the Western Ghats that we have now. And we also know that India separated completely from Seychelles Islands in the 70 million years ago, and it moved northward and it touched the Asian plate. If you were to follow my cursor, this is the Asian plate, and these are the Indian Seychelles complex, and they went northward and they hit it. And then later, what happened was that the oceanic plate of the Arabian Sea subdued into the landmass, thereby further elevating the now Western Ghats that we have. And this is how the Western Ghats are formed, and the Western Ghats are older than the Himalayas because they were there even before the Indian complex hit the Asian plate, right? Which marks the inception of the Himalayas. So that is the reason you would have heard people saying that the Western Ghats is older than the Himalayas. All right, fine. And the next thing that I'll be talking to you about are some peculiar ecosystems that we get to see only here in the Western Ghats and nowhere else in the country. We have few ecosystems of this sort, and if I were to prioritize them in order, then I think Myristica swamps would be of the first priority. And Myristica swamps are basically swampy forests which have major plant representatives from Myristicaceae. Myristicaceae is just the botanical family of nutmeg and many, many forms of other genuses under the family Myristicaceae. And one peculiar thing about this swamp region is you have continuous flow of fresh water ranging somewhere from 5 centimeters to up to 15 centimeters. It can either be a thin water film that is in continuum with the land surface, or it could be a big water column that is continuously flowing, and they are fresh waters, and the air to water and the soil to air interface is completely blocked because of the because of the continual water fill. And it is for this very reasons that plants put out what are called as knee roots, or which is very peculiar to the Myristica swamps. These somewhat function like pneumatophores, in which they increase the surface of roots to the air, but they don't have any lenticels as such that one would see in the pneumatophores of mangroves, right? And where do we have it? We have these Myristica swamps in most of the states that Western Ghats traverses through, and we have a lot of them in Karnataka and Kerala. And in Karnataka, places like Dakshin Kannada and Mangalore and Uttar Kannada will have a lot of these forests, Myristica swamps, basically. And there is a beautiful study that was done by one of my seniors in the lab, and I think all of you all should know it. So there is this species of Myristica called Myristica fatua, and what happens here is they have beautiful tiny little flowers, and they exhibit Beckerian mimicry. Now, Beckerian mimicry is a form of mimicry given out by a German, not a German, he was not a German, an American British botanist called as Herbert Baker. Now, what he says is that this mimicry happens within a species, but it takes place between sexes, which means that this mimicry is a intra-specific yet intersexual mimicry, in which the male flowers or the female flowers mimic each other. What we find from this species is the male flowers are rewarding, which means that they produce a lot of pollen grains and they attract the visitors. In this case, they attract a lot of bees. And what happens is the female flowers, on the other hand, would definitely want the flower visitors loaded with the pollen to come on them, but yet they are very, very cheeky in that they don't want to produce any reward. So they mimic their male counterparts. And honeybees are deceived, and then they go and mount onto the female flowers, hoping to get some pollen reward, but in turn give off some of the pollen that was dusted onto the body. And what is even more interesting is that my senior and my PI ran a model in which they had this honeybee eye color space. Okay, so this honeybee eye color space is just like a hexagon, which tells you that which part that image falls inside this hexagon. And to their shock, they found both the male flowers as well as the female flowers occupied the same loci in the hexagonal plot, telling us that the bee recognizes male and female as the same flower. Okay, and this is a very good example of Bakerian mimicry, and it is even more happy that people have studied it in our own Ghats, not very far from the place that I stay. It was worked out in a place called Kulattapura here in Trivandrum, and this is something that I wanted you to know. And the second interesting thing that people actually have studied is people have studied seed dispersal that happens through freshwater crabs. Now, if you were to see here, you'll see that this is the fruit of Myristica fatua, and this is the black color is actually the seed, whereas this scarlet red one is actually the aril. And it is to this aril that the crabs actually come, and they feed on this aril. In the process of feeding, they are able to transport the seeds for more than 10 meters from the mother plant, which is like a very big distance, given that they are doing this away from the mother plant for better establishment and recruitment in the Myristica swamps. So this is another wonderful study that we have recorded here. And the other plant that I would want you to introduce is this plant is called Semicarpus catlecanensis, and Katlecana is basically a village in Dakshin Kannada, and hence the specific epithet commemorating the name, the place from which it was discovered first in. Now there is a very beautiful, this is again a Myristica swamp dweller, but it is but it is a different genera totally. It doesn't fall inside the Myristica genera. Right, so there is a very little balance, and I don't think if this data is published, but I got to know this from a researcher in CEPF who's in close association with ATRI Bangalore. And what he said was how wonderful a pollinator and a seed dispersal forms a loop in establishing plant populations of this plant. Now, what actually happens here is the flower is pollinated by a butterfly called as the Malabar tree nymph, and the seed is dispersed by the lion-tailed macaque. And what they found out was in plots where there were very low population of both the lion-tailed macaque and the Malabar tree nymph, the populations just did not fruit or flower. They did not fruit or they did not set seeds. And it's quite, and we know the reason why, because they are bereft of the two major uh, animals that would want to deliver two important life processes of a plant, one being pollination and the next one being seed dispersal. And what they did is they created a lot of they upsurged the number of populations of the pollinator and the seed disperser, and then they realized that the fruits had actually took place, and then now they're growing this plant in the nursery and they're re-establishing them back into the Myristica swamps. That is another wonderful work that CEPF was doing in connection with the ATRI. When I was going through and gathering information, I found out that there is yet another wonderful ecosystem, and that is called as Eleocarpus swamps. Now, Eleocarpus is basically the genus of Rudraksha, and these swamps are dominated with the members of Eleocarpus. And in the Western Ghats, we have a lot of Rudraksha trees, and it is known to inhabit these swamps. But nothing much about these swamps are known. But what we do know is we have these patches of Eleocarpus swamps in Karnataka and regions of Kerala, whose information we completely have no avail of, right? And the next ecosystem that I have to talk to you about, and that is almost synonymous with the Western Ghats, is the sholas. And it is not just found in the Western Ghats, it is also found in Sri Lanka. But shola comes from a Tamil word called sole, which actually means orchards. And when you're talking about sholas, many scientists and many ecologists would just refer to these. If you were to follow my cursor, you'll see that they are continuous patches of tropical, stunted, high elevation forests. Now, these are called as sholas, but these make up the shola forests, whereas these continuous grasslands that you see here make up the shola grasslands, basically, or the montane grasslands. And the mountain grassland community has not been studied much, but we do know that there are a large amount of grasses that grow there from um, different species of grasses with different birds are found there. And what we do know is we know that there is a large amount of work that has been done in the shola forests with respect to the plant diversity. And Priya Davidar tells us that this shola forests are usually dominated by the Lauracean members or the cinnamon family plant specimens. And this is all that we know about the shola forests up to 2007. After that, what scientists have done is they have actually studied the orchids, and orchids are abundant in the grasslands as well as the forests of sholas. And what is even more wonderful is the habit of the orchids in both of these types of both of the grasslands as well as the forests. We find orchids which have taken up terrestrial habit more in grasslands, and we find orchids that have taken epiphytic habit in the forests. And Jeevit has also described about 37 species of orchids in the Shola grasslands of Nilgiris. And another thing is there are these bovids called as Nilgiri tars, and these Nilgiri tars are extremely sure-footed, and they are the only mountainous goats that we have here in the Western Ghats, and they have almost taken complete control of Sholas. And if you were to, you would easily spot them in places like Erivikulam and Periyar Tiger Reserve and all the allied Anamudi regions. And this is surely a very beautiful sure-footed mammal that you should set your eyes on when you're walking in the Sholas. And we have and the Shola Islands, also called as the Sky Islands, is very aptly called because these Shola forests are extremely cut off from the rest of the other forest, in that they have a very high elevation and there is no any forest above them. Thereby, they are also called as Sky Islands. We do have a scientist called an ecologist called V.V. Robin in Isar Tirupati, who works on these aspects, but his is more concentrated towards birds and landscape ecology. And what you actually would find inside these forests are these beautiful category of birds that you wouldn't find anywhere outside. Those being the black and orange flycatcher that you have here, and the Nilgiri laughing thrush. And these are the many birds that Dr. Robin also works with, including the Nilgiri Shola kili that he works on. And this is the orchid wealth in the Sholas, and you have humongous of species, and each and every flower communicating to the outside world in its own ways. Right? We just know another interesting thing is we just know that there are these many species, there are these many individuals present, but what we don't know is how they interact with the community and how they actually fit in and what they actually give out to the community that they're residing in. So we don't know anything about their natural history. We don't know anything about their pollination biology. We don't know how they actually establish themselves. No studies have been given out by no such community studies have actually out here. But this again is like a good thing because you know that these orchids are here and you can build on from whatever you have got. There is a lot of animals in the Western Ghats, that is the least to say, but I have just divided the fauna into pollinators and seed dispersal, and I've just taken very little from both of these subtypes because that is all that my slide could actually fit into. And we have a large amount of fauna, like I told you, and since mine was more towards the plant-centric one, I just am giving you a general gist, but this is nowhere equal to the total fauna put together across the Western Ghats. That would be a very, very underestimate. Okay, so what we have here is we have the southern birdwing, which happens to be the state butterfly of Karnataka, and it is visiting the Clerodendron flowers. And this was the flower. This is the Malabar tree nymph. If you remember, I was talking to you about a plant called Semicarpus, and I told you that Malabar tree nymph is the pollinator, and this is actually the plant. And we also have blue mormon. Blue mormon is the state butterfly of Maharashtra, if I'm not wrong, and all of them are high wingspan or large wingspan species, which means they have the ability to fly up to the highest canopy and probe on the flowers there, making them very reliable long height pollinators. And this is the Nilgiri langur, if you're able to follow my cursor. This is the Nilgiri langurs, and one fascinating thing about Nilgiri langurs is people have also found it to be a pollinator for a tree called Culenia, and that is the first recorded therophily we have in the country. Therophily simply meaning mammal pollination. And all of these that you find, the four down here that you find, are potent seed dispersers. We have the giant squirrel, we have the lion-tailed macaque, we have the Indian hornbill or the great hornbill, and we have the brown palm civet. And all of them are potent seed dispersers. I mean, every seed disperser animal has its own broad or a narrow niche of seeds, which actually helps them
Sachin,
to.
sorry to interrupt.
Yep.
Participants, please turn off your camera while you're visualizing the webinar. 20 MCs at 05. Yeah, thank you. Sorry, Sachin,
Yes,
please continue.
that's all right, sir. That's all right. Okay, and there is some. So there are. So where was I? Yeah. So I was talking to you about how every seed disperser has its own broad or narrow niche of seeds, and thereby helping the plant to establish themselves. And the hornbills are also called as the gardeners of the forest, which means that they have the ability to translocate seeds for up to 15 kilometers away from their mother plant in whichever direction that they fly. And that is another wonderful yet breathtaking things that these animals do to help the plant without even they knowing that they're helping the plant, right? And so I was telling you that this is the great hornbill. We also have two other species of hornbill, one called as the Malabar grey hornbill and the other one called as the Malabar pied hornbill. And this Malabar pied hornbill, if you were to Google it, you'll see that it is a white black hornbill. And one specific thing about this hornbill is it feeds on the fruits of a plant called strychnine. Okay, now strychnine is a very poisonous plant, and Malabar pied hornbill is the only hornbill that is known to feed on these fruits. Of course, we don't have any scientific basis to tell this because we don't have any studies that talk about this, but this is all through general observations and through documentary records that we actually know that strychnine has somehow, or Malabar pied hornbill being the only bird to set its beak on the fruits of strychnine, right? Okay, so with this, I'm going to take you to the most exciting part of this talk, which is the plant intelligence. Okay, I'm going to give you 4 case studies, all of them focusing plants. All of these 4 studies have happened here in the Western Ghats. And if there is one thing that I would want you to think in common of all of these 4 case studies put together is the intelligence of plants. How wonderfully they have manipulated and deceived the insects or the anything that visits the plant, and how wonderfully they have taken whole control of them, and they have gotten their job done. Okay, now that's the best part. And this is what is collectively called as plant intelligence. And I'm going to give you 4 case studies uh, for you to understand and appreciate the intelligence of plants. The first thing is called as the first example is protocarnivory and deceptive attraction in 2 varieties of Amorphophallus. Now we all know that Amorphophallus is a very smelly plant in that it gives a very foul stench, to say the least. And this species, especially the Amorphophallus commutatus, scientists, especially Sachin Punekar, is the person who actually told us about this. He was the one to lay scientific basis for this. And this is a single species, Amorphophallus commutatus, but these two are different varieties. The one towards my left is Anmodiensis, and the one towards my right is variety Nov, N-O-V, okay? And what actually is happening here is something called as protocarnivory. Now, what is protocarnivory? Protocarnivory is just like a primitive evolutionary state in which the plant or anything, any organism that has adapted for protocarnivory knows how to attract the insect, knows how to kill the prey. Let us not put it as insects, let us put it as prey. The organism knows how to attract the prey. The organism knows how to kill the prey, but what the organism doesn't know is how to digest and take the nutrients from the prey. Okay, and this is called as protocarnivory, and it's like a very, very evolutionarily um, stemming state for eucarnivory. Okay, we see Nepenthes and Venus flytraps and sundews and all of those. All these are plants adapted for eucarnivory in that they are able to attract, kill, and digest the prey and use up the nutrients. But this is like a tiny little, just a budding stage of carnivory, and that is termed as protocarnivory. Now, what happens here actually is that the flower actually blooms in the evening, and it gives out a very foul smelling stench to which there are many beetles attracted. If you were to see, you see that there are a lot of beetles that are attracted and they fall into the spathe. Now, if I can quickly brush through the flower structure because it looks very complex, this is a family of arums, right? All Amorphophallus belongs to a family called Araceae, and they have a specific, a rather specialized inflorescence. It is called as spadix. Okay. Now, what is spadix? Spadix has two components. One is it has the flower stalk, which is this basically, and the flower stalk is nothing but flowers. You have male flowers here. Follow my cursor. If not, you will be lost. These are male flowers that are on the lower side, and these are female flowers onto the upper side. And on top of this, that is the brown color stuff that you see all over here, is nothing, it's just appendix. And what appendix does is it produces either oils or odor to just attract the visitors towards it. All right, and this is a spike in which the flowers don't have a flower stalk, and they are adorned with a extremely decorative bract, which is called a spathe. All right. So this entire thing is what is called as a spadix, and this is called as the spadix cup. So this is exactly where the beetles go and fall. They come, they fly all the way, they sit on the appendix, they get marred, and they are extremely attracted, and they fall into this. And after they fall into this, what happens is that that beetle would have got some pollen because it is definitely coming from another flower, and on the way when it is going down it brushes it off onto the female flowers and it falls down. Okay, and if you observe, there are a lot of warts here that don't allow the beetle to go upside, or rather they are caged inside. Okay, and this happens overnight, and overnight is when anthesis happens and the flowers break and the male flowers anthers break open and a lot of pollen mass is shed. And a very peculiar thing is the next day, there are a few set of beetles that are able to escape out, which are called as the bold beetles. And there are few set of beetles that are not able to go out, but are rather inside and they die. Those are called the weak beetles. Okay. And then the next day when the flower completely wilts away and when scientists break open and cut open the spathe, they find a lot of beetles stuck towards the inner side. Okay, and we also know from phylogenetic analysis that carnivory has independently diverged in many clades of Araceae. Okay, and rightly pointing out that this protocarnivory could be an evolutionary buds point from which eucarnivory would have actually evolved. Right? So this is the first case study that we talk about protocarnivory. And the second one is called deceptive attraction in another variety of the same species called as the variety Nove. What happens here is the plant, this we all know that in this appendix, there is a type of a semiochemical. Now, what is a semiochemical? A semiochemical is basically a pheromone, or it is basically a volatile that is produced by an organism to direct very specific information to the receiver. Okay, and the semiochemical that the plant is giving is basically the sex pheromone of the beetles itself. So the beetle gets very excited because they are smelling this in the air. They come onto the appendix, they go down, and they know that the volatile is even more downside in the spathe cup, and they are sure that they find mate. They are sure that they're going to find a mate there, and lot of beetles congregate, and they congregate, they come, and in the process what happens, they get dusted with a lot of with a lot of pollen on them, and then they mate with their mates, and then they fly off. The plant is not doing anything here. The plant is not giving the rewards, the plant is not giving pollen, the plant is not giving nectar, the plant is not giving floral tissue, but it is just deceiving the pollinators to come take their gametes, and in the process copulate, and they leave the flower to yet another flower, right? And this is another very good way of attraction, deceptive attraction rather, in this species of Amorphophallus, in this variety of Amorphophallus commutatus. Okay, fine. And the next thing, the next scenario is called as the double deception that we find in Ceropegias. Ceropegias belong to the family Apocynaceae, and they are a very good fly traps. Okay, and what they actually do is something very, very beautiful, and something that requires extreme intelligence. Okay, and this is called as honeybee under attack. So this mechanism is called as honeybee under attack. What happens now is there is a honeybee, okay, and when the honeybee is killed, or rather when it is dying, it emits a cocktail of chemicals, okay. And what happens at that time is there is this specific species of fly. It is a kleptoparasite. Now, what is a kleptoparasite? It is nothing but an organism that feeds on the food of another organisms. Okay, so there is an organism A that has killed a honeybee, and the honeybee is emanating this complex mix of volatile into the air, and the fly gets hold onto this volatile, and it comes and feeds on the honeybee. There are two ways in which the flies can do. They can come alone and they can share this food with the actual predator, or they can congregate and they can take the whole honeybee carcass to another place. All right, and in doing so they get satiated, and then their jobs are done without investing time on hunting, or without investing time on killing something that could potentially kill them. Okay, recollect the sting in the honeybee, which is enough to kill a fly. So the plant actually emanates this, and the plant actually emanates this honeybee type of volatile mix, and thereby deceives the fly to come inside. And once when they come inside, they get inside, and this is the gynostemial cup. And what happens here is there are a lot of downward pointing arrow which ensures smooth getting in, but the exit is very, very difficult because all these trichomes are pointing downward. And there are two ways in which the plant deceives the housefly. First thing, it is emanating something, it is emanating a potential food for the fly, and the fly is coming inside, but there is no food. Deception number one. Deception number two is, in spite of the fact that the fly stays here for quite some time, there is no nectar produced by this plant, and there is just pollen that is dehisced, and that is dehisced on the fly, and the fly doesn't get any reward for taking up this pollination. So that is deception number two. And what we don't know is how the fly actually the fly is definitely weakened, right? Because it is there for the whole night or for many hours, and the fly is definitely weakened, but we don't know how efficient they are in transferring the pollen dusted onto them. But that is something that we should look at. This was first demonstrated by Anne-Marie Hayduk in the University of Bayreuth, Germany, when she was a PhD students there, and she calls it the honeybee under attack. And India has about 55 species of Ceropegia, and 98 of them are endemic to the Ghats, 4 of which I have shown here. And we have no idea about the pollination biology of them. And we don't even know if honeybee under attack actually takes place in our land, in our Ghats, or if there is something else that the plant are potentially giving out. So that is something that we have to still work on. And the third one is called as the mass. So this happened in Kars Plateau by some, and the work was uptaken by some German scientists. And what they actually found out was these 3 species of Urticularia. And Urticularia are basically insectivorous plants in which they have urticles down in their roots. And these urticles actually have a tiny little opening, and they have the ability to suck water inside, right? So when they are sucking water continuously inside, anything that comes with the water, it could be zooplanktons, it could be beetle larvas, it could be anything that can fit the orifice of the urticle, it gets inside and they digest them and they uptake it inside their body. That is why they're called bladderworts, because of the bladder that is present. All of these happens underside of the plant in the water, in the roots, but above them are these beautiful flowers, uh, who no way seem that they will kill insects and eat. And they are now here again, these flowers want insects, but they don't want to kill them, but they want them to uh, convene them with their important life process called pollination. And they mass flower, okay? And what scientists have actually concluded is this mass flowering, how it actually enhances the reproductive success in these bladderworts. And what people have actually found out is that the mass flowering actually helps multiple attractiveness, which means that these flowers are extremely small. They'll be 1 to 1.5 centimeters across diameter, and it will be very difficult if they were to flower individually in sparse location, okay? Because that wouldn't have such a heightened attractiveness, and there are many a times that the flower visitors would have actually missed on it, okay? And mass flowering actually helps in cumulative attractiveness, if that is the right word to say. And the next thing is, the plant, each flower does not have to invest on a lot of rewards. For example, if one flower here is producing say 20 microliters of nectar, if there are a lot of flowers, there are the lot of flowers can produce say 5 or 7 microliters of nectar and not 20, because it lowers their investment. That is one way to see it. And the second way is, a plant when it produces 20 microliters, a flower when it produces 20 microliters of nectar, and the crop capacity of the honeybee, if it were to be 20%, then the single honeybee that comes will just feed on one flower and get back to its hive, put out the nectar and come back. In that process, we don't know where the pollen is actually going. But if there were to be a tiny little reward, if you were to decrease that nectar amount, then the honeybee won't be satiated, so it will visit more flowers. And it lowers the reward, it lowers your investment as a flower, and it also increases the visitation rates, because that is what promotes cross pollination, right? And that is those are two things. And the next thing is, it probably increases floral constancy. When a flower visitor know, okay, this is the color, this is the shape, this is the nectar guide, this is the area that I should look for. And if a flower is getting rewards from these across different several bouts, then there are higher chances that they can actually become constant to these particular flowers, thereby increasing efficient and clean cross pollination, right? And it is a perfect strategy for small flowers, and these being small flowers have intelligently took this up. All right, this was another thing that I wanted to share. And the last thing is, in the case study part, is the prey pollinator conflict in Drosera species. Drosera, if you were to know, if you remember, it was an insectivorous plant, and they don't have any pitchers like Nepenthes, or they don't have laminal blades like Venus flytrap. But what they all have is, they have exceptionally long trap mucilage hair that are actually thigmotrophic, and they have a highly tactile influence on the insect that visits them. They get all these mucilage hairs together, and then they digest them with the digestive droplet on each of these mucilage hair. But what there is a question that scientists that has always lured scientists, and that is, you need insects. The plant need insects for two things. One, as a food, that is prey, and the second is to carry out pollination and thereby pollinator. And how do these insectivorous plant actually get into and use their brains? I mean, they don't have brains, plants don't have brains, but still they are intelligent. That's another fun part. And how they actually have used their intelligence, and they have reduced this prey pollinator conflicts. And people have studied this in Drosera. And there are two things that they have done. Like in this species, you see that Drosera burmani, it is spatially separating the flowers from the trap, which means that the insects that will come to the trap are very less likely to visit the flower. Or rather, the insects that are going to visit the flower are less likely to visit the trap because they're spatially separated. And the other thing that scientists have actually understood is not all species take up spatial separation. There are some species that have very closely arranged flower traps and insect traps and flowers. So in that case, what is going to happen? There is no spatial separation seen there, but what scientists have actually found out is these traps and flowers have different volatile cocktails or odor cocktails that can actually selectively call pollinators and insects for the traps. That is the best part. And when scientists have ran behavioral analysis for the flower visitors, they found out that all the flower visitors only showed response to the floral odor, but whereas the insects that came for the trap visited both the flowers as well as the traps. And these components of these volatile cocktails are very specific, and this is one of my favorite studies that I have ever come across till date. Fine. With this, I think I will give a gist of my research work, what I do for a PhD. Basically, I work on flowers. I work on a lot of flowers. I work on plant community. I do a community level work in which I ask the question about how different floral traits, right? Now, floral traits are nothing but characters of a flower. It could be anything. It could be color, it could be scent, it could be nectar guide, it could be the symmetry, it could be shape, it could be anything inside a flower. And I ask what floral traits act as harness signals to indicate rewards and flowers, and in a community level. So which flower trait can actually honestly indicate reward status in flowers? In rewards, I mean pollen or nectar. And another part of my PhD deals with flower visitor networks. So it's basically like a food web, but here you don't deal with trophic levels as such, but you just deal with flowers and the visitors. How many visitors visit different flowers and how they appear as a network is what I study. And I also study about how diverse, or rather how different are these networks in a monsoon and a dry season, right? Apart from that, I also do take up very tiny little projects, and I work on some critically endangered plants and understanding why they have very low distribution and understanding the plant communities. And these are the tiny many things that I do here in the lab. And with this, I think I have completed 40 minutes, and that is what I also intended to do for my presentation. And I would like to leave you with this quote telling that one of my favorite quotes from Jane Goodall, that is, the greatest danger to our future is apathy. We all know that there are different things that are continuously hitting onto it could be the Western Ghats, it could be the um, Indo-Burma region, like the previous talk, like the previous speaker spoke. And I'm pretty sure that the speakers tomorrow also will have one thing to say in common, that our biodiversity hotspots are in the verge of getting ruined. Okay, but but the best thing that you could do as future scientists is to actually study them in such a way that you can get out effective measures to actually protect them, because you cannot protect them uh, when you know nothing about them, right? So we need to have strong evidence data, because before even which we can know, and that could actually help in a better conservation. I mean, they talk in a landscape level, but I talk in a plant specific, animal specific, organism level, in which I think each organism has its own way of leading a life in this world. And if you have to conserve it, you should know the life process better, because without which I don't think any measures of conservation will just dwindle down. And that is the take home message that I want you to go with. And this is the plant that I'm working on. It is a species of Lectranthus, and it is supposed to be critically endangered. And we have also found an extended distribution of this here in the Agastya Malay. And this is my email ID. If you ever are excited about the works that we do here, you can anytime write to me, and we can get in touch. Okay, and I think with that I've done. It was very crisp, but I'm pretty sure I've kept up my.
Yes, Sachin. Thank you. Thank you for accepting our invitation and being here. Another dynamics, how vegetation, flora, floral diversity, plant insect interaction are also crucial components of biodiversity hotspots, more so close to us here, the Western Ghats in South. And a very
Yes,
interesting talk there from Lal Mond Sangha from the Indo-Burma.
Indo-Burma.
We had two talks, two biodiversity hotspots. Yeah, over to Selva Priya for the moderation.
Thank you so much, sir, for such a wonderful presentation.
You're
I can
welcome.
say with I can say with the certainty that you were successful in making all of us fall in love with the plants. We truly weren't aware of such details about the plant species you spoke about. We were
Thank
able
you.
to look at pollinators, dispersal topics, Myristica swamps plants, and plant intelligence topics, sir. We have learned a lot with in a completely fresh perspective, sir. Thank you so much, sir.
Thank you very much.
Over to you, Mathli.
That was one interesting and informative evening. I truly thank Lalmon Sangha sir and Sachin sir for taking out time from their busy schedules and sharing with us such a valuable part of their research, for informing us about some of the most extraordinary hotspots and about innumerable species of several plants and animals from distant regions like Myanmar, Meghalaya, Mizoram, and the Western Ghats that have been discovered and many more that are yet to be discovered. I can say with certainty that everyone present here today was fascinated and amazed by your talks just as much as I was. Your passion and dedication is truly admirable. I would like to wish you the best of luck for all your future research and endeavors. Thank you.
Thank you. Same to all of you
Thank
all
you.
too.
Thank you, Sachin. Thank you, Maithili. Thank you, Abhishek, Adina, all of you, the team behind this national webinar on biodiversity hotspots of India. The first day is done. Tomorrow we have two more presentations on the Himalayas by Dr. Mehreen and on the Sunda land, more of the Andaman Nicobar by Dr. Sahu. So with that, we are ending today's webinar, the first day of the national webinar. There are things put up on the chat for the participants. Please fill in the feedback form that is going to fetch your participation certificate. Secondly, also look into our links of the National Science Association, the Instagram account, the YouTube channel, and the Facebook account. Please enroll and follow them because that's where you're going to get all these videos uploaded and information related to these webinars, all those that were conducted in the past and also those that will be conducted in the future. There again, you have it on the screen. So with that, we are winding up today's session. Thank you all. Thanks to both Lal Monsanga and Sachin for their presentations on the first two biodiversity hotspots, the two more for tomorrow. Thank you all. Good night. Take care. Yeah, Abhishek, you can stop the recording and then we can wind up.